Hey everyone, welcome to your quad for Hershey and Chase. The Hershey-Chase experiment was very important, and it's one that we studied because it defined genetic information as nucleic acids. So before, proteins and nucleic acids were kind of battling to see which one was the genetic information for the cell. And so Hershey and Chase was the experiment that kind of closed the lid and definitively defined nucleic acids as the genetic material. So what we're going to see is an overview of their experiment. If you can kind of think of this experiment in two phases, separated by the line drawn here, what they did is they used radioactive isotopes to identify what was being passed on from viruses to bacteria. Now, the type of viruses that they use is known as a bacteriophage, which is shown right here. Now, it was known that bacteriophage had proteins and nucleic acids and really nothing else. The question was, when these bacteriophage infect bacteria, what is it that's being passed on to the bacteria itself? They knew that bacteria would eventually start making copies of the viruses. But what was it that was being passed on by the viruses to the cells in order to encode the genetic information to make more viruses? So in the first part of their experiment, they used radioactive sulfur as the tag, and in the second part of the experiment, they used radioactive phosphate because they knew that these two elements were unique to proteins and to nucleic acids. They could not use CHO or N. Both of these are common in both these macromolecules. So carbon, hydrogen, oxygen are common to both of them. They wanted to use something that would label the macromolecule uniquely. And some amino acids contain sulfur. And so they knew that if they used radioactive sulfur, that only the proteins would be labeled. And only the nucleic acids contain that phosphate group. No proteins contain that phosphate group. So they knew that if they labeled it with the radioactive phosphate group, that only the nucleic acids would be labeled. So using this radioactively labeled protein, they allowed the phage, you can see the phage here, they allowed the phage to inject the genetic material into the bacterium, and then they removed the outer shell of the viruses. When the scientists came in and scanned the bacteria that now had the genetic information of the viruses, they noticed that none of it was radioactive. When they then ran the exact same experiment using the same phage and the same bacteria, only this time what they used is they used a radioactive phosphorus, so a radioactive P. And then again, they allowed the bacteriophage to inject the bacteria. They removed that outer shell, and when they scanned the bacteria afterwards, they found the radioactive tracer was inside the cell. That means that the viruses had passed on the nucleic acids into the bacteria. Now, the big conclusion, once again, of this experiment is that DNA, not proteins, was the genetic material passed on to the next generation. Thanks for watching.